Hello and welcome to this week's What Were They Thinking? Among the news items we have for you this week is continued evidence that infection with COVID-19 has long-term and significant side effects that are likely to present a greater emerging health threat than COVID currently has. New ways to develop and modify stem cells to create transplantable tissues. The genealogy of humans, and then bizarre news about octopus farming. These news items, along with others, can be found at the timestamps given below. Let's start with the risk of cardiovascular disease as a consequence of infection with COVID-19. The research comes out of Washington University School of Medicine and is very recently published. It looked at about 20 pre-existing cardiovascular disease outcomes over a year, but they also looked at 3 different groups for this. All of the groups were former USA military, or in other words, veterans. The data came from approximately 1,255 healthcare facilities and over 153,000 veterans. These were the ones who tested positive for COVID-19. There was also a cohort of 5.6 million veterans who did not test positive for COVID, and 5.9 million veterans from 2017 as a control group who had no way of ever being exposed to COVID at that time. The researchers looked at things like stroke, transient ischemic attack, sinus tachycardia, ventricular arrhythmias, pericarditis, myocarditis, angina, heart failure, and more. The simple summary of what they found is that over the 12-month period after infection, there was a huge increase in all of the diagnoses. Some as high as 63% greater risk of developing the diseases in question. This is something we've mentioned in the past. There is a very good chance that due to the nature of the immune response, the tissues and organs targeted by the COVID-19 virus with the ACE2 receptor and a variety of other factors, infection on a widespread scale will lead to extreme, substantial, and incredibly bad health complications downstream. That is, in the next 5 to 10 years, we would expect to see a huge increase in the number of people who are coming to hospitals with complications arising from infection with COVID-19. While still on the topic of COVID-19, researchers have been trying to figure out just how it does what it does. And one way they do this is to create a very simplified version of the coronavirus in question, then study how it responds to various stimuli. The researchers have been based in both the UK and Germany for this study, and what they were trying to figure out is, in a lab scenario, what does the virus do that makes it hard for the immune system to get a hold of it and destroy it? By doing this and using a very simplified version, they avoid many of the issues around creating something that could cause an epidemic. One of the observed phenomena was that the spike that is being used both for developing vaccines and to allow the virus to get into host cells to be replicated behaves differently when exposed to the immune system. When antibodies bind to it, the region of the spike protein folds and changes its conformation. When this happens, the area that it's binding to no longer has the right structure, and so it's that much more difficult for the antibody to remain there and for the immune system to use that antigen site as a means of identifying it as a pathogenic substance or pathogenically infected cell. Speaking of vaccines, Canada has done something weird, unusual, and different. This is Canada in a nutshell some of the time, but in this case, what they've done is they've approved a vaccine made by growing markers from plants, putting these into a shot, and then injecting them into a human. Clinical trials demonstrated that this generated an immune response, and the government took this on board and has now approved the vaccine. The vaccine was developed by Medicago, a Canadian-based company. Where this vaccine differs from others, to a certain extent, is that it does not contain any genetic material, just antigens that the immune system can find and be able to use to develop an immunity to the virus infection. They're virus-like particles, but no virus genome at all. This is a technology very similar to what was used to create vaccines for Ebola, which was grown in tobacco plants. Unlike some of the currently circulating vaccines, this has a higher efficacy against new variants of the COVID-19 virus. 71% with the original strain and 75% against the Delta variant. The only noteworthy distinction between this vaccine and others that are currently available in Canada is this vaccine is only available for those between the ages of 18 and 64. Going now from COVID-19 and all sorts of viral news to stem cell news, and the idea of turning stem cells into bone. Bone can be very important, especially if you are in a major accident where your bones have been broken, limbs have been removed, or there's simply damage that can't be dealt with any other way. Consider what would happen if your bones in your legs or arms were shattered. There is very little option when it comes to transplanting bones from a donor, and for that reason, most people more or less are completely out of luck. This is where stem cells and this particular development could come in useful. It's the ability to regrow a limb, starting with stem cells that will create the bone for an individual. This is a step further along and a more complicated version of past research that used the mineral matrix of bone, had all of the biological content removed, and then had new cells added to it to regrow from an existing bone matrix that was only mineral content. This could completely regrow a limb starting from the bone with someone's own stem cells. That by itself is pretty damn good. The even more impressive element to this is that they were using sound to be able to get the stem cells to turn into bone. Further to being able to turn the stem cells into bone, it is a relatively low maintenance task. It takes about 5 days to stimulate the stem cells, and you only stimulate the stem cells for 10 minutes each day. This means that you could, in theory, be able to regrow bone or develop other material from bones, such as the red blood cell stem cells, using this technique. The advantages of this is that the cells in question don't undergo a lot of the stress that would be caused by using chemical processes. The chemical stimulants and signals that are often used for this kind of research can be quite toxic, and they can generate other complications that can damage the cells. It's been one of the big considerations in just how you make sure the cells you culture behave the way you want them to and don't go rogue somewhere down the track. For example, turning into cancerous cells. Such processes are often very labor-intensive, expensive, and need specialized knowledge. The development here has none of those disadvantages at present. Going from developing modern treatments for long-term disorders that humans have dealt with forever to something of our own history. In this case, a study that tried to amalgamate all sorts of data about human DNA and try and create a rather massive human family tree looking at the various lineages of people across the globe. The work came out of the Oxford University's Big Data Institute, and it used 3,600 individual sequences from more than 215 populations. By combining all of these into one big data set, They could then begin figuring out how everything interacted. This created the branches and roots of the tree. Relying purely on modern human genomes wouldn't have been anywhere near as helpful. Instead, they also relied on Neanderthal genomes, Denisovian, and a family who lived in Siberia about 4,000 years ago. This gives a big idea as to the spread of different populations over the globe, and how at different times Homo sapiens have interacted with them, notably the Neanderthal and Denisovian genomes, which have their own quirky distributions. Moving on to animal-related news, and it's actually about octopuses. And not just octopuses in and of themselves, but the plan for a farm. Octopuses are a surprisingly large food group for many people. There is a very big push to be able to farm them commercially in order to be able to meet increasing demand. We already do something similar with salmon, crabs, other shellfish, and even certain fish species. Octopuses, on the other hand, haven't really ever come into that category, and the theory is that if we can farm this species along with others, we could help to reduce the strain on areas that are overfished. The problem is the interesting ethical concerns that have been raised around doing just this. You may not be familiar with why there would be concerns. After all, as said, we happen to farm many different species, and aquaculture farming isn't that weird an idea. The problem with it is that octopuses are notably fussy about what they will and won't eat, nearly always choosing to eat something that's living over something that's not. This can include others of their own species. That problem is exacerbated when they're in a confined space and they become aggressive. They're also easily stressed in that sort of situation and can engage in self-mutilation. Admittedly, some octopuses do this anyway when they detach their penis tentacle and throw it at the female, literally telling them to themselves. The other issue with it is that, as you may or may not have seen in the past with viral videos, octopuses are very good at getting out of captivity. This means that even if you do have a large farming setup, your actual return on investment may be very small. The other ethical aspect to this is that people argue octopuses are intelligent enough to deserve greater protection than other species. As such, farming them, and farming them with the limitations we've mentioned already, could be seen as cruel and unusual. Going from animal news to space news, but also back to humans. Humans themselves as a species haven't been into space for all that long, and very few have actually been to space. This is why knowing what's happening inside the brain of astronauts now, before space tourism, among other things, really kicks off, is so very, very important. It would appear that travelling in space has at least some effect on neurons and how they're wired in the brain. This research is based on a very small study size, so that is an important caveat to remember. But there are also only a small number of people who have been to space. So it's a case of you take what you can get. The 12 astronauts we're talking about had their brains scanned through diffused magnetic resonance imaging. That is, they get a dye and the dye is put into their bloodstream and it enters the brain, and then you run your MRI. This provides a baseline reading. Then the astronauts went to space, and they then spent approximately 170 days there. When they returned to Earth, they were scanned again. The first noteworthy changes were in parts of the brain responsible for sensory and motor function. This should honestly come as no surprise, but having confirmation is always good. The fact that you are in a low-gravity environment with very different physical activities and needs means the body has to get a different grasp about what it's doing and how it's doing it. There was also dilation in parts of the brain that provide the necessary oxygen and nutrition, the ventricles. These caused slight dilation changes to occur, and this had impact on the corpus callosum. It resulted in initial research that they thought indicated that the corpus callosum had been affected, but it turns out that no, it was the dilation of these ventricles. Our final piece of news for you this week, and also space-related, is the odd structures found on the moon by the Chinese Cosmonautical Agency's U-2 rover. They claim to have found glass spheres. These small spheres appear to be simply sitting there. And although glass isn't uncommon on the moon, consider that it has a large amount of silicon and is regularly hit by large heavy asteroids that are full of heat when they arrive, you can see why glass might be made. It's the fact that these are spherical that is a little bit unusual. It is possible that in the past there was volcanic activity on the moon, especially after it was initially formed. This, along with something like, say, meteorites, could all have resulted in the generation of liquid glass. Liquid glass would have gone up, but it would also have eventually come down, even in the low-gravity environment of the moon. This would have resulted in the sphere-like structures we see. Such a approach was used for a very long time by humans to create lead shot as a round ball, and is even used today for creating something like buckshot for shotguns. Sadly, it is likely to be that simple an explanation, so anybody who's already jumped to the conclusion that it's evidence of aliens or some other advanced society is going to be sorely disappointed. That's all the news we have for you this week. Thank you for watching. If you have found it interesting, consider liking, sharing, and subscribing. Please paste any comments, questions, or suggestions that you have below.